Hello, welcome back to our series of lectures about muscle. In this video, we're going to be covering the topic of excitation contraction coupling. Okay, so first off, I just want to start by saying now that we have gone over some of the basic anatomy of the muscle, I want to talk about how we go about carrying out muscle contraction, particularly on a molecular level. So in this video and the next couple videos, I will be talking about the molecular events that cause skeletal muscle contraction to happen, okay? Just a reminder, I want to remind you that the signal for a skeletal muscle to contract comes from a somatic motor neuron. Okay, so this is the same figure that I used in the efferent lectures, but we're going to be sending an action potential down this motor neuron. That motor neuron is going to send a signal to the muscle to contract, okay? And the process of excitation contraction coupling is basically the process by which action potentials are translated into muscle contraction. So let's take a look at those events. Okay, so first of all, just to orient you, what you're looking at here is this is a motor neuron, like kind of the end of a motor neuron. Okay, it's meeting up with this muscle fiber here. This red part is the cell membrane or sarcolemma of the muscle fiber. This yellow part is the cytosol, also known as the sarcoplasm of the muscle fiber. You can see this T tubule here, okay, this invagination of the sarcolemma. You can also see the sarcoplasmic reticulum here. And inside the sarcoplasmic reticulum is a whole bunch of calcium. So the sarcoplasmic reticulum is acting as a calcium store for the muscle fiber, okay? And then there are a bunch of other things down here, the thin and thick filaments. That's where the contraction is actually going to happen. But let's take a look at the series of events that happen to trigger that muscle contraction, okay? So first off, again, remember, we start with an action potential. Sorry. We start with an action potential coming down the motor neuron. That's going to trigger release of neurotransmitter the same way you would in any other neuron, okay? The neurotransmitter here, if you remember from our efferent division lectures, is going to be acetylcholine, okay? All right, that's this slide. Moving on. The acetylcholine is going to bind to acetylcholine receptors on the skeletal muscle. And again, if you remember from our efferent division lectures, the receptor here is a nicotinic receptor, okay? The nicotinic receptor is a receptor channel, which means that it's a receptor that's also a channel, right? So as soon as, well, not as soon as, but when the acetylcholine binds to the nicotinic receptor, it's going to cause that receptor, which is also a channel, to open, okay? When that opens, that's going to allow sodium to come into the cell, into the muscle cell, the muscle fiber, okay? When we have ion movement across the membrane, hopefully you are aware by now that that's going to lead to a change in membrane potential, okay? So we're gonna get a depolarization. It's gonna be a graded potential, kind of similar, it basically is an EPSP, except that in the muscle, we have a different name for it, which is the EPP, the end plate potential, okay? So we're going to trigger an end plate potential, a graded potential in the muscle fiber. That end plate potential is generally going to be like, it's pretty reliable, it's pretty big. So usually when you have synaptic transmission here, you have an end plate potential that's big enough to bring that muscle fiber to threshold, and then you get an action potential, okay? And that action potential is going to travel along that sarcolemma, okay? So it's going to travel down the T tubule. The next thing that happens is, remember that an action potential is a wave of depolarization, right? So we're depolarizing the cell membrane, okay? When that depolarization goes down the T tubule, it is going to encounter this molecule right here, which is called the DHP receptor, okay? So DHP stands for dihydropyridine L-type calcium channel. Don't need to worry about that really, but DHP receptor, okay? DHP receptors are voltage sensitive. So that means that if you depolarize the membrane around a DHP receptor, it's going to change conformation, okay? It also happens that this DHP receptor is mechanically coupled to this calcium channel that is in the sarcoplasmic reticulum, okay? So when we depolarize the DHP receptor, we get a conformational change that's coupled to that calcium channel. It's going to cause that calcium channel to open, and that is going to allow calcium to be released from the sarcoplasmic reticulum into the cytosol or the sarcoplasm, okay? All right, and then finally, we have muscle contraction. So basically the presence of calcium is going to allow a series of molecular events to occur that leads to the movement of the thick filament relative to the thin filament, and that's going to cause the muscle to contract. We're going to go over these events in the next videos, but this is kind of, what you should know for now is that we have calcium coming in, that's going to trigger muscle contraction. Okay. Oh, right. Last thing that I want to mention here. In order for the muscle to relax, we need to kind of erase all of these signals, right? So after we get this signal, calcium will eventually be pumped back into the SR, the sarcoplasmic reticulum. Okay. And finally, I just want to point out that I am going to have you watch this video that shows the events of excitation contraction coupling. I do want to point out there is some terminology introduced in there that I haven't introduced to you. Don't worry about it. I won't hold you responsible for knowing it, but it's a very nice animation of the whole process.